All right, background of the DNA molecule and slide number 2. And down here it has a comment for it, so hold off on the comment until I get through talking about the slide here. Um, you can see way back in 1865, a guy named Gregor Mendel kicked off all of this stuff about DNA. Now, did he talk about DNA specifically? And the answer is no. What he talked about was terms that he called factors. And factors to him were uh, something that the male parent and the female parent gave to their offspring to create the new offspring. So that he was trying to figure out why this particular pea plant and this pea plant, when they were mated, gave them this kind of pea plant. And he called all those things factors. We have changed that name now. Instead of a factor, we call them genes. So we currently now call them genes. Uh, but from that point on, it was basically a question of where are these genes? Where are they located? Are they located in DNA? Who knows? At that point in time, they didn't even know about DNA. It wasn't until 1869 when Mitscher came along and basically discovered this stuff called nuclein. That's what he called it. He didn't know the structure was deoxyribose nucleic acids, and that's where we coined the term DNA for the sugar and the nitrogenous bases in there. When we say nitrogenous bases, that just means a molecule that has nitrogen, nitrogenous base. And the D stands for deoxyribose. Okay, deoxyribose. Anything with OSE on the end of it is a sugar. So that's deoxyribose nucleic acid. The only thing DNA does not have in its initials there is the phosphate. Otherwise, it would describe the whole molecule setup. But from this point on, there were two groups of scientists. One group uh, knew for a fact that genes were located in proteins that are located in the cell membrane. And they believed this because proteins they knew from past experiences did most of, if not everything, in the cell. If the cell needed to get something done, it was done by proteins. And they pretty much knew that even way back in this time frame. So that was a pretty good guess. And at this point, they're all guesses. Nobody at that point had any hard data to back up their guesses. So I couldn't call it a theory yet. And if they did, they were a little premature. With the other group of scientists, they believe that it must be contained in this stuff called DNA. Why? They couldn't tell you, other than there's a huge organelle that contains a lot of stuff. It is one of the largest structures in the cell, and it, for all intents and purposes, does nothing. And they couldn't understand why a big, huge organelle would be sitting inside of a cell and do absolutely nothing. And therefore, they believe that it doesn't do nothing. It must do something. Therefore, that something could be that's where the genes are located. But they couldn't prove it. These guys in here did different setups and different experimentations, which helped lead to further discoveries. So Mueller, where there's X-ray induced mutations, they started playing around. If you did this, you did this, you could change the sequence of what's going on. And we know now that the X-rays would induce mutations to the DNA molecule itself and not necessarily what's going on. They knew the DNA was being messed with, but or not DNA, the genes were being messed with. But again, they still didn't know where they were. They didn't know whether it was the proteins embedded in the cell membrane or it was something in the DNA. So the battle still went on. So Griffith tried to figure this out. And he goes, all right, I've got an experiment that will prove once and for all its genes are located in the DNA. And so he sets up this experiment. And it was a really grandiose experiment. Seems kind of simplistic when we look back on it, but from 1928, it would have been a really good, solid experiment. It fell a little short, though, of the mark. And this is why the battle continued on until Hershey Chase. In 1952, Hershey Chase basically put the lid on the coffin. 1953, Watson and Crick nailed it shut. So we'll go through the Hershey Chase experiment. We'll go through what Watson and Crick did, and the rest over here, the genome sequencing project at Cold Harbor Springs, that's in New York. What it was all about, we'll talk about that later. So what do I want you to put down here for a comment? And that would be that the timeline from 1865 all the way to 1953, there was a large debate as to where the genes were actually located. Are they located, or were they located in the DNA, which they knew very little about, or was it located in the proteins that are embedded in the cell membrane? Okay. Like I said, that slide would take me a while to do, and that was almost 7 minutes. All right, slide 3. This slide here, I'm just looking for the name of the actual scientist that figured this out. So this was in 1869, extracted phosphorus-rich material from the nuclei of a human white blood cell, and he named the material nuclein. Why white blood cells? Why not just blood? And the reason is there's 3 main components to blood. There's plasma, which is just the liquid. There is red blood cells, but a mature red blood cell does not have a nucleus. No nucleus, no DNA. So hence, why take it from red blood cells? You have to have immature red blood cells, and they're far fewer than mature cells. So red blood cells would be much harder to get any kind of DNA from it. White blood cells, on the other hand, is just like a normal body cell. It has a nucleus, contains DNA. You can extract the DNA from there with very little problem. Okay, so again, to get this guy's name, you would go back to the little chart here, look up 1869, and you'll find that it's probably somewhere in this region. And you just put in the guy's name. This one here, Pyrimidines and Purines. Okay, this is purines and pyrimidines is the generic name for a structure of a molecule. And I won't go into that structure, but we'll say that purines there's 2 molecules that have to do with DNA, and that is adenine and guanine are purines. They have a certain structure. Pyrimidines are thymine and cytosine. And a gentleman by the name of Chargaff, this would be this guy here, in 1950, he spent, 5 to 10 years figuring out the process that would tell him that there is a relative relationship between these two, where about the same percentage of adenine would be found in the same percentage of thymine. So when these two are found together, they would be in about the same percentage. When guanine and cytosine were found together, they were also in about the same percentage. Now, he never said that these two were equal. And when you look at this, the double line here between A and T does not mean equal. Okay, so let me see if I can blow this up a little bit. So you can see here that A equals T. No, that is not meaning A equals T. What this means is adenine double bonds with thymine. I added another line here because this timeline did not have it properly done. This means cytosine is triple bonded to guanine, not double bonded. It had another equal sign here, and that's not true. And so I think on yours, I wasn't able to put the line. I didn't notice that it wasn't there until after I'd already started talking about it. So if you want, you could add another line in there. You just go up to your line tool, draw a little line, boom, you got it. Okay, just note that cytosine triple covalent bonds with guanine. Adenine double covalent bonds with thymine. Okay, it's one of the reasons why thymine is more susceptible to sunlight. It's only double bonded to adenine. And it's also the molecule itself is more susceptible to UV sunlight. So when we go back to here, what is actually going on in here? Well, this is saying that adenine, or you would say in a comment, because of Chargaff's rule, or just Chargaff's rule states that adenine bonds with thymine and guanine bonds with cytosine. That's what would go in here. Chargaff's rule states that adenine bonds with thymine, guanine bonds with cytosine. Okay? All right. So moving on here, and I need to erase some of my stuff. Get rid of these so you guys can figure out, whoa, not that. I want the picture. There we go. Okay, so you can see we still have DNA molecule. It's unwound. The purple lines represents the backbones, and the backbones are made of phosphorus, or phosphate, not phosphorus, phosphates. So phosphate molecule is bonded to a sugar, which is bonded to a base, which is then, depending upon the base, with cytosine, it's triple bonded to guanine, which is bonded to a sugar, which is bonded to a phosphate. Okay? So if I was to draw this out, okay, so there's a phosphate. Whoops. And it's bonded. Wow. There we go. It's bonded to that sugar. It's bonded to this sugar. All right. And if I take another phosphate, and then another line, that would be bonded to this sugar. And this one's bonded to this sugar. So this thing goes sugar bonded to a phosphate, bonded to a sugar, bonded to a phosphate, bonded to a sugar, bonded to a phosphate, bonded to a sugar. So what were my boxes that I had up there before? Well, these are called, or those were called nucleotides. So a nucleotide is, and I'll draw a box around it again, one phosphate, one sugar, one base. So down here in the comments, put that a nucleotide equals a phosphate, sugar, and a base. And that is a DNA monomer. A string of these monomers makes a polymer. So that would be the DNA polymer, which is all of the nucleotides put together. But a monomer of DNA is one nucleotide. Okay?